So we're going to cover two chapters. We're going to cover the cell cycle, which includes mitosis, and then the next chapter, which is meiosis. Now, the goals of both of these, the goal of mitosis is cell generation, regeneration, growth, and asexual reproduction. And so that is going to be asexual reproduction would be single-celled organisms. And in multicellular organisms, we also go through mitosis, and that's going to be like our somatic cells, our body cells, so that we can have like recreate our skin cells and our blood cells. And so that gives us cell regeneration and also gives us growth. Now, meiosis is going to be the production of gametes, and that is only in sexual reproduction. Now, I have a video here. Of course, you can tell I like Paul Anderson with Bozeman Science. So he will have a short video of where he kind of goes through the cell cycle, mitosis, and meiosis.
Hi, it's Mr. Anderson. Welcome to Biology Essentials Video 28. This is on the cell cycle, mitosis, and meiosis. In other words, how we go from one cell to all the cells, the trillions of cells inside our body. Meiosis is important because that's how we make sex cells. Now, let me digress a little bit. This is a whiptail lizard from the desert Southwest, and what's interesting about this is that it's a female lizard, and when it wants to make more lizards, it will simply use mitosis to make an exact copy of a cell inside its body. It's called parthenogenesis, or virgin birth, and it will make a brand new number of baby lizards, and they're all females. So they don't have males. It's rare to not have males. It's rare to not have meiosis, and the reason why is that all the whiptail lizards are genetically the same, but it works. And if you live in a fairly stable environment, it tends to work. Now, we're not going to go into the specifics of mitosis and meiosis. You can look, I've got videos on each of those that talks about the different phases. What I want to talk about this is, in general, what do mitosis and meiosis do, and how does the cell cycle work, and how is it controlled? And so a diploid cell is going to be a typical human cell, or a typical cell inside an organism. It's going to be 2N, and what that means is it has two complete sets of chromosomes. So for example, in humans, 2N equals 46. That means we have 46 pair of chromosomes. And so the goal of the cell cycle in mitosis is to make a copy of that cell. In other words, to make a diploid cell. That diploid cell can enter into the cell cycle again, make more cells and more cells and more cells. And so the way that we make new cells in our body or replace cells that are damaged is mitosis. In meiosis, we're going to take a normal diploid cell, and we're actually going to make a haploid cell. We're going to make sperm and egg. And so in humans, N now equals 23. Now, if we were to just stop there, we wouldn't have diploid cells anymore. But fertilization, where egg meets sperm, is going to combine those two cells to make a diploid cell. And now that diploid cell can enter into the cell cycle again. And so we've got kind of two loops. We've got the mitosis loop and the meiosis loop. Mitosis is used to make all the cells in our body. Meiosis just makes gametes, or sex cells. I also, in this video, want to talk about how we control the creation of diploid cells, so it may be better to come from here, and how we use cyclins. An example of that would be the mitosis promoting factor to control the cell cycle and where it is and where it's headed next. Now, if you're talking about cell cycle, the best place to watch is start is with videos of actually cells dividing. And so this is a cell undergoing division. So we start with one cell, and it makes an exact copy of itself. Now, when you're watching it, let me go back a second. On these first two videos, what you don't see is everything that actually happens before the cell makes a copy of itself. In other words, before the cell is actually able to divide, it has to duplicate all of the DNA and all the machinery of the cell. This last one, we're looking actually inside the cell itself. And so the cell division actually has two parts to it. Part one is going to be the division of the nucleus. And we call that, in general, mitosis. And then after we've divided the nucleus, you'll see the chromosomes actually separate here. Then we actually have the division of the rest of it, and that's called cytokinesis. Cytokinesis is the break apart of all the other parts of the cell. So the machinery of the cell, the mitochondria, cytoplasm, things like that. And so let me see if I can start that up. So first of all, there we go. first we have the division of the nucleus. You can see the chromosomes pulling apart, and then you have cytokinesis, or the division of the cell itself. And so all mitosis is one cell forming two cells, and those two cells are identical to that first cell. And that's how we go from that first fertilized egg inside us to the trillions of cells that we have inside an adult body. So when you're replacing cells in your body, you're doing it through mitosis. So let's look at the cell cycle. And so what happens is you'll have a cell enter into this. So a cell is going to look like this. It's going to enter it into the cell cycle as one cell. Let me make a little better arrow. And then it's going to eventually exit out as two cells. Each of those cells, remember, could go back into the cell cycle. And so this is how we make all the cells in our body. Now, I've heard that a lot of the dust in a room actually are dead skin cells. And this is what skin looks like. So skin is going to be created new cells, and then they're going to migrate up to the surface, and then we're eventually going to lose skin cells at the top. But we keep replacing those cells, and to do that we use the cell cycle. So let's look at the parts of the cell cycle. If we start first with that cell entering in, the first thing that it'll do is it'll actually enter into the G1 phase. G1 phase, the cell is actually going to grow. It's going to get bigger and bigger and bigger and bigger. We then enter into the S phase. During the S phase, we're going to actually, using DNA replication, we're going to copy all of the DNA inside a cell. It then goes into the G2, or the growth 2 phase, where it continues to grow and gets ready for division of the actual cell. And so G1, S, and G2 are all part of what's called interphase. And if you look at a cell, it's generally in interphase. It's growing, it's copying its DNA, it's growing, or maybe it's just working. It's doing what a normal cell does. And so most of the life of a cell is in interphase. It's in these three, G1, S, and G2. And the actual copying of the nucleus and copying of the cell, this mitotic phase, is actually going to be really, really small. If it never divides again, it'll actually stall out in something called the G0 phase. And so we've got cells inside our body. You've heard of this, maybe cells of the central nervous system, muscle cells, for example, that never copy themselves during your whole lifetime. They're in what's called the G0 phase, or they're just waiting, and they're not going to make divisions. And so how does a cell know when it's time to divide, and when it should go on and when it shouldn't? The best analogy I can think of is it works kind of like an hourglass clock. And so there are little proteins inside a cell, and as those proteins accumulate throughout the life of the cell, eventually you get a critical number of these proteins at the bottom. And once we have enough of those, then it actually tells the cell to advance to the next stage. And those proteins are called cyclins. And so let's look at what cyclins look like. Cyclin, I'll represent it here. It's simply as a protein. But if we look here at the G1 phase, the S phase, the G2 phase, and mitosis, there's a set of cyclins, or a set of these different types of proteins. And what they're going to do is they're going to build up. And so cyclin A and B are ones that I'm really familiar with. Cyclin A will actually build up during the S phase, and then it'll drop off right as the cell divides, as it goes into this mitotic phase. And so cyclins will actually accumulate. And so those are like the sands through an hourglass. They're going to get more and more and more and more cyclins as the cell goes through the cell cycle. Now, the other chemical that I want to talk about is something called CDK. CDK is simply a cyclin-dependent kinase, and a kinase is simply going to be a chemical that can speed up actions within a cell. And so CDKs are found in all living organisms, and you can actually take CDKs from a yeast and put them in our cell, and they work just as fine. So they show homology through evolution. And so cyclin-dependent kinase, if you look at their name, are simply dependent upon cyclin. So I made a little animation of how that works. And so a typical cell in your body is going to have a bunch of CDKs in it, or cyclin-dependent kinases. And so we could say this is like right here. But throughout the life of the cell is going to start building up and accumulating larger amounts of cyclin. So the amount of cyclin is going to get larger and larger and larger. So eventually what happens is the cyclin is going to fit into the cyclin-dependent kinases. Now we have an activated CDK cyclin complex. What does that mean? We have something, a protein that's able to do things. So now you can think of, we've like mustered this army and now the army is ready to do something. And so what does it do? Well, we're right here, so we're just about to enter into the M phase. So we're just about to do mitosis. And so what these cyclin-dependent kinases do is they act on the cell itself. So they are a specific type of CDK is called the mitosis promoting factor, or MPF. And what that does is when it builds up enough of these cyclin-dependent kinases, they're actually going to work on the cell. So one thing they'll do is they'll actually break apart the nucleus. So we were able to start dividing that cell. Another thing that CDKs will do is they'll actually work on the microtubules that build this spindle. And so all of these together will work on pushing that cell into the mitotic phase, or into this next step of the cell cycle. The neat thing about each of these is that after they've actually done that, they'll actually gobble themselves up, they'll disappear, and then the whole cycle begins over again. Okay, so if we kind of talk big picture about what happens in the cell cycle, a typical cell right here is going to be, let's say this is a typical cell. A typical cell right here is actually going to be 2N. It's diploid. You have one chromosome from mom, one chromosome from dad. So in this case, it's going to be 2N equals 2. It's then going to duplicate itself. So during the S phase, it's going to make copies of itself. And so at this point right here, we'd actually have a 4N cell. It's made copies of that. And at this point, we can either take the path of mitosis or the path of meiosis. And so in the path of mitosis, that'll simply split in half, and now we'll have two 2N cells. And if you look at these two 2N cells, they're exactly the same as that first cell. So this is what's happening to the chromosomes. If we look at that 4N cell, and as it goes into mitosis, it'll actually line up, it'll split in half, and then it'll split in half again. And so what you have is actually N cells. Those are called haploid cells, and we have 4 of those in meiosis. And so the cell cycle will take a diploid cell, make 2 diploid cells in mitosis, or make 4 haploid cells in meiosis. That got a little messy, so let's look at it in a little bit more detail. So mitosis is how we replace cells in the body. So cells that are broken, cells that are broken down, cells that we need to replace, we do that through mitosis. Or when we want to grow, how do we go from a very small organism to a very large organism? It's just making more cells. And so this would be to make it simple, we're going to start with a simple cell. This is going to be a 2N, it's diploid, but 2N equals 2. So this would be that first parent cell. It'll then duplicate all of the DNA. So now we have this characteristic shape. It has 2 sides to it, and this side and that side are copies. That's what happened during the S phase. They'll then meet in the middle. They'll divide in half. So this would be our mitosis phase, and now we have 2 diploid cells. And if you look at those cells, they're exactly the same as that original cell. And each of those are 2N equals 2. So that's mitosis. What happens to these 2N cells? Well, they can enter into the cell cycle again, and it goes over and over and over again. In meiosis, what happens is a little more detailed. Remember, meiosis only deals with making sex cells, or in reproduction. And so in this case, to make it to show you meiosis, I had to increase the number of cells. In this case, or chromosomes, 2N equals 4. In other words, we have 2 chromosomes from mom, 2 chromosomes from dad. So they'll copy themselves. So now we actually have a 4N cell at this point. But since we're doing meiosis, this is where actually crossing over occurs. And so parts of this chromosome will swap places with this chromosome and vice versa. What that gives us is variability in all of the sperm in the egg. They'll divide in half. Now we have 2N cells, and then eventually we'll have N cells equals 2. And so we started with 2N equals 4, now we have N equals 2. Each of these 4 things in a male become a sperm, and in a female, one of them will become an egg, and the other ones will actually form what are called polar bodies. And so what do we get after meiosis? Well, we get sperm and we get egg. And we can then go from these original 2N equals 2 to eventually, or N equals 2, to a 2N equals 4 zygote. And so now we're back to a diploid cell, and that diploid cell could enter into the cell cycle. This is a zygote to make more cells, and eventually make sex cells to make the next generation. So that's cell cycle, mitosis, meiosis, and I hope that's helpful.